Hello, this is our AP Biology project, and it's a claymation on genetics. Here, DNA has a double helix shape. This was initially discovered by Rosalind Franklin while working in the lab of Mars Wilkins by using an X-ray crystallography to take pictures of DNA. However, the credit for the discovery goes to Watson and Crick, who, when seeing the image, noticed that it was DNA being the double helix shape. While Franklin knew the certain object was a double helix shape, she did not know it was DNA. Replication has a few important proteins that contribute to the replication process. DNA replication is a semi-conservative process where one of the parent is always in the new DNA. The first enzyme would be helicase, which unzips the parental double helix so that the other proteins can come in. Then there would be the primase, which synthesizes the RNA primer at the 5' end of the leading strand, and in the lagging strand, it makes the primers where the Okazaki fragments are. DNA polymerase then comes in and synthesizes a new DNA strand by adding nucleotides from a 3' to 5' action to the leading strand, and from the lagging strand it does the same but goes from each primer to the next, creating Okazaki fragments. DNA polymerase 1 removes the RNA primers and replaces them with the nucleotides. Ligase then comes in and glues together the DNA wherever the Okazaki fragments are. Now you can watch the process occurring. There is the helicase coming in and unwinding the DNA. Now that that happened, RNA primer comes in and attaches to the end. After that, DNA polymerase 3 comes in and attaches its nucleotides to the leading strand, and on the lagging strand it's a little more complicated. The RNA primers come in certain spots. Then DNA polymerase 3 comes in and starts putting the nucleotides. After that, DNA polymerase 1 comes in and replaces the RNA primers. Now there are Okazaki fragments. Ligase then comes in and glues the two fragments together to create the nucleotide. This process will continue for the rest of the DNA. Next, we'll be talking about the central dogma. So we just showed you replication. Now we're going to be showing you transcription and later on translation. Transcription takes place in the nucleus. When RNA polymerase pries the two strands apart, it then attaches to the promoter region where it recognizes the Tau box. The transcription factors then join the polymerase where they start the initiation of transcription. In the elongation step, the polymerase goes down the line and adds the nucleotides, adenine binds to uracil instead of thymine. Finally, once polymerase reaches the end of the termination signal, it is removed from the DNA, and transcription ends when the mRNA detaches and the 5' cap and the poly A tail attach to the mRNA. These spliceosomes then cut the introns, and the exons are the ones expressed. Ligase then attaches the exons together, and now the mRNA leaves the nucleus and enters the cytoplasm. Once RNA enters the cytoplasm, it meets the ribosome to start translation. Initiation is the first stage of translation. In order for it to begin, mRNA needs to attach to the A site with the AUG codon so it can start. Then elongation starts, where the mRNA goes down the chain to the P site of the tRNA, and it will hold the correct amino acids from the A site to the P site. The P site will attach more amino acids to form a chain. Once the mRNA is done, it will go down to the E site where the mRNA exits the ribosome. This can only happen if the mRNA has the stop codons. Then the polypeptide chain detaches from the P site, and chaperone proteins will fold the chain to make actual proteins. Next, we're going to be talking about alternative splicing. This is a normal splicing where the spliceosome cut out the introns and leave the exons, which are the coded regions that are expressed. The introns are the blue and the exons are the pink. Alternative RNA splicing is when exons and introns are switched, and this is one way protein synthesis is regulated. Genes can code for many polypeptides in which it depends on what is supposed to be the exon. Different nucleotides are transcribed and translated depending on what the exons are. Finally, we're going to talk about point mutation. Point mutations are mutations that only affect a single base pair of a gene. A really good example is sickle cell anemia. It happens in the base pair where it codes for the B globin polypeptide of hemoglobin. This gene results in a mutation because of an abnormal protein because of a base change. Thank you for watching our Claymation project.